Hi, my name is Bianca, and today I'll be presenting about extending the bounds of non-invasive neuromodulatory treatments using a technique called low-intensity transcranial focus ultrasound sonication. I work at the Department of UCLA Psychiatry, and my mentors are Dr. Susan Buchheimer and Dr. Taylor Kuhn. Currently, neuromodulatory treatments today that are popular are surface structure treatments and neurosurgical treatments. Surface structure treatments include transcranial magnetic stimulation, transcranial direct current stimulation, electroconvulsive therapy, etc. And neurosurgical methods include deep brain stimulation. And these methods, while great, are problematic because many of the disorders, such as anxiety, that we would like to treat have primary target regions in deeper regions of the brain that are currently inaccessible because of the surface nature of these methods that are available. So the most popular methods are DBS and TBS. And DBS, while highly efficacious, is difficult to access because it requires neurosurgery, which is its own adventure itself, and also is difficult to access because it's financially and physically difficult. TMS, on the other hand, is unpredictable in terms of its efficacy, and it has minimal risk, which is a great benefit in lieu of DBS. Ideally, a neuromodulation technique combining the efficacy of deep brain surgery and the minimal risk of transcranial magnetic stimulation would be great. However, this is not possible at the moment, but that's what I'm here to present about. Transcranial focused ultrasound sonication, or TFUS, is the size of a hockey puck, and it is targeted, effective, and non-invasive. TFUS is, neuromodulation researchers are calling TFUS the holy grail of brain stimulation treatment because it is cheap, painless, and portable, and can focally modulate a specific region in the brain, and only that region in the brain. And as such, it is what they're saying is the next big thing. TFUS thus presents itself as one of the most exciting and potentially ideal devices to enable treatments that require penetrating neural tissue and underlying deep brain structures, most especially for compromised populations, such that who cannot physically or financially afford the costs that current alternative treatments require. By eradicating regional limitations of non-invasive neurostimulation, TFUS offers a potentially promising alternative as the future's next effective neuromodulation treatment method for neurological diseases and psychiatric conditions such as anxiety and Alzheimer's. TFUS is based on ultrasound, and ultrasound waves are acoustic wave energies with frequencies higher than the human ear can hear. And it can penetrate the skull to reach and exert an effect on deep neural tissue with a high spatial resolution. TFUS, like other neuromodulation techniques, can be applied to psychiatric and neurologic patients and provides advantages over pharmacologic treatments, including more focal and direct impact on neural circuits. Importantly, TFUS appears to be safe for neuromodulation in humans based on the current literature and is generally well tolerated, including, unlike high-intensity ultrasound, TFUS acts via a non-damaging, non-thermal mechanism without any negative impact on histology or the blood-brain barrier. So to study this, we asked, can TFUS selectively increase regional cerebral blood perfusion via targeted neuromodulation? And our hypothesis was that yes, TFUS treatment will result in an increase in cerebral blood flow perfusion in targeted regions. So to do this, we targeted the amygdala and the entorhinal cortex. And our hope is that TFUS will potentially increase cerebral perfusion and thus also increase emotional regulation and downregulate amygdala function. In the entorhinal cortex as a target, our hope is that targeting the entorhinal cortex with TFUS will increase perfusion and thus also directly enhance working memory. So, to study this, we designed a randomized, double-blind, within-subject crossover study. We asked our subjects to come in for 2 visits, 2 weeks apart, one visit targeting target 1, or the amygdala, and the other visit targeting target 2, while the other target served as the control. So our sample was a great sample ratio with half male and half female, with a total of 6 subjects, and our mean age was 61.8 years of age with a standard deviation of 8.25 years. Before and after each TFUS treatment, we administered a series of neuropsychology evaluations, as well as a psychophysiological task. The TFUS treatment was targeting the amygdala and the entorhinal cortex. TFUS targeting was made possible using the Siemens 3T Prisma MRI scanner. And TFUS is MRI compatible, MRI conditional. And we used an MRI guided placement protocol by Norman Spivak and Taylor Kuhn to guide our transducer. So the transducer, again, as you remember, is the size of a hockey puck. And so right here, that white dot is the transducer. And the red circled regions are the amygdala, the right amygdala and the left entorhinal cortex that we targeted. And the subjects served as their own controls. And so we targeted the amygdala and the entorhinal cortex on different days. And because the TFUS technology is MRI conditional, we were able to use the MRI to visualize where in the brain exactly we were targeting in live time. These were our TFUS parameters, and this is what a TFUS pulsation beam looks like, up really, really close, of course. To collect data, we collected arterial spin labeling images, or ASL, taken before and after the TFUS treatment. And we created difference photos using FSL software to correct for motion, partial volume effects, and normalization, and registered this to standard space. ASL difference photos were then overlaid on the subject's high-resolution T1 weighted images. And our results were exciting. ASL shows selective regional perfusion at TFUS-targeted regions. And you can see this by the red, which is the right amygdala, and the left entorhinal cortex. And so when we targeted the amygdala only increased in perfusion. And when we targeted the entorhinal cortex only increased in perfusion. And so the amygdala here, when we targeted the amygdala, again, the entorhinal cortex, which our target was over here, and the entorhinal cortex would be located down below the amygdala, it didn't increase in perfusion. And vice versa for the entorhinal cortex. And I repeat this because it is so exciting, because that means that it suggests that TFUS treatment can regionally select and neuromodulate specific regions in the brain. And that's our hope, at least. And so future directions, well, for future directions, of course, this was a pilot study. And so we are planning to continue our study with larger sample sizes. And we are currently conducting research on the relationship between TFUS and functional connectivity, and potentially how TFUS can impact behavior, memory, and emotion regulation. Little work has been done on TFUS neuromodulation in humans, and we'd like to point this out because our results are very new and in this extremely new field. So our results are potentially exciting because it suggests that TFUS neuromodulatory treatments could hopefully one day help treat anxiety by targeting the amygdala and even Alzheimer's disease by working to directly enhance working memory. With the safety, accessibility, and non-invasiveness of ultrasound via TFUS, TFUS is a promising technique for neuromodulation with a huge patient population potential. And so we are excited to share this with you. And these are my references. And I'd like to thank you all for listening, and especially my mentors for making this possible, as well as our lab and our collaborators. Thank you.
Hi, I'm Piergiorgio Salvan, and I'm going to talk to you about the optogenetic stimulation of the mouse entorhinal cortex and how this reshapes whole brain dynamics. So the authors declare no competing financial interests. So brain network communications arises as an organic property from the interaction of billions of cells linked together in interconnected circuits. This is possible because the transient synchronization of neuronal spiking is believed to be the medium of this information transfer. And this is a ubiquitous property in the brain, both within and between neuronal populations. Now, despite this being such a key aspect of brain communication and also of cognitive function, the mechanisms or the neural coding underlying flexible and selective communication has been unknown. So in this work, we asked how local activity can be selectively and flexibly routed across the brain network. And to answer this question, we used optogenetic concurrent fMRI stimulation. So this is a little cartoon showing off our experimental design and computational approach. And as you can see on the left, we used, again, this concurrent opto-fMRI approach in which we stimulate in a block design fashion the excitatory neurons of the entorhinal cortex on top of the basal activity to discharge at frequencies of 5, 10 Hertz, so within the theta range, and at 20 Hertz, so in the beta range. We then used hidden Markov modeling as our computational approach to co-model whole brain fluctuations in the bold signal, both in terms of amplitude and covariance, meaning, again, bold amplitude and functional connectivity. This resulted in a finite set of states that are transient and repeating in time, and each of them is characterized by a unique parameter of brain activity and of functional connectivity. Now, what I'm plotting here is just a cartoon, is the output actually of the hidden Markov models. And I'm plotting with each color a different state, and the amplitude of the color represents the active state at each. As you can see, align the stimulation with the optogenetic stimulation, and as you can see, there are bursts of activity in relation to this optogenetic stimulation. And this is super interesting because the HMM is an unsupervised approach. Therefore, no information about the stimulation was fed in the decomposition method. And so this allowed to basically detect transient states that were evoked by the neuronal spiking. So in order to answer our question, we first asked whether does single area neuronal spiking give rise to multiple states of network dynamics? And so to answer this question, we perform a t-test between the opto-degenerate group, so the group expressing photoreceptors versus control. And we find here, you can see the results of the t-test expressed in -log p-values. And you can see that there were 3 different states that increased activity compared to controls during the optogenetic stimulation. And the most important thing is that both of these all these 3 states were co-activated during stimulation, and they were characterized by different or distinct spatial maps, both in terms of amplitude and functional connectivity. Now, if you were to use a normal GLM approach and model the activation to the stimulation, what you will find is just one single pattern of functional activity. So now that we have established that inducing EC neuronal spiking causes distinct networks dynamics to emerge, we can ask some more fundamental questions about the mechanisms or the neural coding underlying selective communication. And as we know, the entorhinal hippocampal system is very well known to be modulated within the theta rhythms, and this represents, again, the basis of the EC hippocampal system dynamics for communication, and they are thought to support memory. So what we've asked here is what is the effect of modulating local neuronal spiking in the entorhinal cortex with theta frequencies? And in order to do this, we tested a repeated measure ANOVA, testing for the effect of 5 and 10 hertz. So the theta frequencies on the HMM activation probabilities. And what we found is that the F-test provided a positive answer for 2 out of the 3 states being active during stimulation. But what is even more interesting is that when we unpacked the results in terms of the individual t-test, what we find is that whilst state 10 responded more responded preferentially at 10 hertz stimulation, state 11 responded preferentially at 5 hertz stimulation, meaning that by modulating the stimulation frequencies, it is possible to achieve different communication channels or different brain network dynamics. Now, what these results provide is that basically we are showing that frequency modulation of neuronal activity is a sufficient mechanism to enable selective brain network communication. Now, what is important about the EC hippocampal system is that it is not just well-studied and well-known, but it is also the key component that is known to go wrongly during Alzheimer's disease, for instance. And so in order to provide some insight of the crucial role of this mechanism into behavior, we started a transgenic model of Alzheimer's disease. And we asked, how is selective network communication through theta modulation impaired in Alzheimer's disease? And to do this, we performed a mixed design ANOVA, where we tested the effects of frequency by group, therefore frequency by group interaction. And what we found is that 2 out of the 3 states responding to optogenetic stimulation were also showing an interaction of group by frequency. And again, we then unpacked the T statistics, and what we found is that whilst the wild type mice had shown this this pattern of this dissociation in their response, in the AD mice we find no evidence for this dissociation, and indeed state 11 showed no preferences for either frequency, whereas state 10 responded at both 5 and 10 hertz. Now, what these results are showing together is that the frequency modulation of the entorhinal cortex neuronal activity is impaired in a model of Alzheimer's disease. Now, just to conclude, we set up our research question that was, how can local activity be selectively routed across the brain network? And we provide evidence that distinct network dynamics or information streams can be achieved by theta modulation of the entorhinal cortex neuronal activity. This basically is a causal evidence that selective brain network communication can be achieved through a multiplex neuronal code. And what is most important, perhaps, for translational studies is that by showing that this mechanism of selective communication being altered in the Alzheimer's disease, we might provide some evidence of how the aberrant modulation of network dynamics might underline cognitive decline. And with this, I would like to thank all my co-authors, the founding bodies, and all of you for your attention.
Good morning, everyone. I'm Shile from Chen Institutional Center for Translational Research in Neuroimaging and Data Science. Now I will introduce our work. Electroconvulsive therapy treatment responsive multimodal brain networks. Here is the outline of my talk. Firstly, background. Major depression is the leading cause of disability worldwide. Though depression is typically treated with different forms of psychotherapy or pharmacotherapy, ECT is regarded as the most effective treatment for severe and treatment-resistant depressive episodes for both major depression and bipolar disorders. Cumulative studies report that ECT is significantly more effective than pharmacotherapy, with 50% to 60% of patients achieving rapid remission of depression after an ECT course, compared with 10 to 40% with either pharmacotherapy or psychotherapy. ECT is also particularly effective in patients with major depressive disorder with psychotic features and in elderly adults with MDD. Despite the efficacy of ECT, the neurobiological underpinnings and the mechanisms underlying symptom improvement of ECT-induced antidepressant response remains unclear. Identifying ECT-responsive biomarkers could clarify the mechanisms of antidepressant action and promote individually tailored and precision treatment. Although ECT results in significant clinical improvement, patients often show cognitive difficulties during the ECT series. For example, hippocampal-dependent cognitive functions, such as declarative memory, are most adversely affected by ECT. Specific brain regions, including the hippocampus, amygdala, as well as the whole brain structural changes, were investigated following ECT. Two recent mega-analyses of structural imaging investigations confirmed that ECT induced broadly gray matter volume increase, including hippocampus, but neither of them have found association with treatment outcome. So this result suggests that the efficacy of ECT is unexplained by hippocampal enlargement or single gray matter modality, which alone might not serve as a variable biomarker for treatment outcome. And the focus on a specific brain region or single imaging modality may limit discovery of structural and functional brain changes in a whole point view that are related to clinical outcomes. Therefore, this investigation aimed to address this limitation by multimodal fusion. Here, firstly, we want to identify the brain structural and functional remodeling associated with reduction in depression severity after ECT. And second, whether the identified treatment responsive networks remain group difference between healthy controls. And third, compare the identified ECT responsive brain networks between responders and non-responders. And finally, the relationship between ECT responsive brain networks with memory. This study includes 118 patients and 60 healthy controls recruited from the University of New Mexico and the University of Los Angeles. Here, note that 76% patients were non-medicated and 79% of patients were right unilateral. According to the 4 goals we stated before, we performed the following corresponding 4 analyses. Firstly, HDRS total scores were used as a reference to guide a 2-way MRI fusion for all the depressive patients. And second, back reconstruction was performed to compare group difference between patients and controls of the identified ECT networks. And third, HDRS guided fusion was performed on responders and non-responders subgroups separately. And fourth, correlation between all the identified treatment responsive network and memory was calculated. One joint component was identified that's longitudinally discriminated pre- and post-ECT and negatively correlated with HDRS within pre- and post groups. We can see that after ECT series, decreased brain activity in prefrontal, insular, hippocampus were accompanied with increased gray matter volume in ACC, caudate, thalamus, medial temporal gyrus, insular, and hippocampus in SMI. Note that hippocampus and insular were identified within both SMI and fMRI treatment responsive networks. And we found that the identified ECT responsive networks preserved group difference between patients and controls for both fMRI and SMRI brain maps in both pre- and post-ECT groups. We also performed the same HDRS guided file for gray matter fusion separately on responder and non-responder subgroups to extract specific ECT responsive brain networks. The responder and non-responder groups have unique treatment responsive networks. For responders, the ECT series was associated with decreased brain activity in the prefrontal cortex and increased gray matter volume in caudate, thalamus, and hippocampus. While for non-responders, the ECT series was associated with decreased brain activity in medial temporal cortex and thalamus, and increased gray matter volume in left insular. And interestingly, the identified responder and non-responder specific covariant file and gray matter brain networks can be replicated under remitter versus non-remitter separation criteria, in which the most replicated brain areas for remitters and responders are prefrontal in FILE and caudate and thalamus hippocampus in SMI. For non-responders and and non-remitters. The most replicated brain regions are medial temporal gyrus and left insular in gray matter. So, comparing with non-responders, the responders' specific ECT responsive networks are mainly located in the frontal limbic circuit. We also tested the correlation between all the identified ECT responsive multimodal brain networks with memory scores, and we found that there is no significant relationships were observed for the ECT responsive networks and verbal memory in all patients, responder and non-responder subgroups. To the best of our knowledge, this is the first attempt to estimate ECT treatment responsive biomarkers by jointly analyzing two types of MR data under the guidance of the HDRS total score. Our investigation demonstrated that ECT treatment response was associated with brain alterations at multimodal level, with reduced FIO in prefrontal, insular, and hippocampus, and increased gray matter volume in ACC, insular, thalamus, cortex, medial temporal cortex, and hippocampus. Second, when the subgroup were analyzed separately, ECT responsive brain networks have different patterns in responders, frontal limbic, and non-responders, insular and medial temporal lobe. And third, they identified ECT responsive networks in all patients, responders and non-responders were unrelated with memory performance. So collectively, this study indicated that treatment responsive and ECT-mediated neurocognitive impairment may be related to distinct circuitry. And the treatment responsive frontal limbic network, although broadly distributed, could be optimal for targeted engagement by neurostimulation therapies. Finally, I would like to thank my supervisors, Professor Calhoun and Professor Sui, for funding me doing this research. Further information is available in the related posters and papers. Thank you for your attention.
So first, I would like to thank the organization committee for giving me the opportunity to present today. So today I will tell you about the entrainment of theta oscillations with visual rhythmic stimulations in order to boost auditory working memory in humans. Several studies in brain stimulations are trying to optimize brain stimulation protocols. And one way of doing that is to use information-based neuromodulation procedures that target specific neural networks and also that aim to target specific neural dynamics, so mainly the brain oscillations that are rhythmic fluctuations of brain activity. So the idea is to determine how the different nodes of a given network that is involved in a given cognitive task are communicating with each other. To date, the main studies using these approaches have used rhythmic TMS or TSES. Today, I just wanted to introduce that we can design information-based neuromodulation protocols by using visual rhythmic stimulations. So first, I will start with the model of interest. So the cognitive modality I used, that is auditory memory with a simple recognition task. So participants are asked to encode a stimuli, can be an auditory, melodies, words, or other type of auditory stimuli. And this encoding phase involves mainly auditory regions bilaterally. Then they have to maintain the information during a silent retention period and to keep this information in memory. And this involves a more distributed network with both ventral and dorsal regions. Then they have to retrieve this information, so compare what was kept in memory to a stimuli coming to the auditory system. We decided to use this model because the three different phases of the task are well defined, and the neural networks involved for each phase are well defined as well. But also, with only one paradigm like this, we can investigate two types of memory. Short-term memory, for which you just keep the information in memory, and working memory, for which you keep information, but you also have to manipulate this information in memory. So just to illustrate the two different tasks, so as I mentioned before, for short-term memory, you just compare two auditory stimuli. But for working memory, here in this example, for a given working memory task, the participant listens to the first melody, then the second melody is always reversed. So 1, 2, 3, 3, 2, 1. So that the first tone of the second melody corresponds to the last tone of the first melody. And then the participants, during the silent retention period, have to mentally manipulate the information and reverse the first melody to be able to perform the task. When you contrast those two types of memory using an fMRI recording or other types of neuroimaging approaches, we can see a clear pattern in the dorsal pathway involving both bilateral and inferior parietal sulcus, SMA, and prefrontal regions. So it means that we have a specific network that is related when you have to manipulate information in memory. So with our general idea of defining information-based protocols and targeting specific neural networks, if we want to boost manipulation abilities during working memory, we will target specifically for each participant this dorsal pathway. Then we also have to identify the neural dynamics based on oscillations. So to do so, we can do some recordings of electrophysiology with MEG or EEG. And when you do the same task and you contrast working memory versus short-term memory in this dorsal pathway, you can observe a clear pattern of theta activity during the retention period, so activity around 5 Hz. And what was interesting in this previous study is that theta power and also the functional connectivity at theta between different nodes of this dorsal pathway were predicting participants' behavioral performance for the working memory task. So when they were asked to manipulate the information. So in order to test the causal relationship between those theta oscillations and participants' behavior, we just stimulate them for each trial using TMS in this previous study. So 5 pulses in 1 second for each trial. And we observed a clear relationship between the oscillatory entrainment, so the way how well we were able to boost theta oscillations in the dorsal pathway, and the behavioral benefits. So now, using this previous literature, we can say that we have a specific neural network to target, the dorsal pathway, specific metrics of oscillations, theta activity, and we also know that the different nodes of this brain network are communicating with each other at theta. So based on that, now we'll try to define our visual stimulation protocols that aim to target this network at this specific frequency. And here we want to benefit from the fact that the dorsal pathway, also called the central executive network of the multiple domain system, is not only involved when you have to manipulate auditory information in memory, but it's also involved when you have to do a lot of different cognitive tasks, such as quantitative calculations or visual mental rotation. So what we wanted to test here in this study is, as we are working on a network that is really multiple and that is involved for multiple different tasks, can we use visual stimulation to prepare the network working at the appropriate frequency before the memory trials? So to do so, we use the fast periodic visual stimulation by presenting to the participants Shepard's forms that are flickering on the screen at 5 Hz. And in this condition, the participant will have a feeling of rotation, will see the object rotating. Our hypothesis is that just by perceiving this, we might recruit or involve mainly the dorsal visual pathway. So going from the visual to the parietal and the frontal regions. And the control condition here would be the presentation of stimuli that are random with no feeling of rotation. And this type of stimulation should involve mainly the ventral stream. So that's what we tested in the first experiment with an MEG experiment in 8 participants. We just asked them to passively observe these two types of stimulation, so random and rotating forms. And we observed that when the participants were stimulated with rotating objects, it recruits visual areas, parietal areas, but also frontal regions. While when they were stimulated with only random objects, it was mainly visual regions. So here the metric we are observing is the stimulation brain phase locking, so meaning what are the brain regions that are in phase, that really follow the stimulation. And when you do a direct contrast between rotating and random, you observe a clear pattern of entrainment in several nodes of this dorsal pathway that are left IPS, right IPS, and left dorsolateral prefrontal cortex. So based on that, the idea is, can we prepare the network before each trial of a working memory task using visual stimulation? So the task we are using is the same I was describing before, but with just shorter melodies. So the participants listen to a first melody of 3 tones, 1, 2, 3. Then the second melody is always reversed, 3, 2, 1. And they have to detect a small change in the altered condition. First, with an EEG recording in 23 participants, we just confirmed that theta activity, so theta power and theta phase locking in the parietal network and in the dorsal pathway are predicting participants' behavioral performance, as it is illustrated on the figure here, during the retention phase. So when the participants are manipulating information. And this was illustrated as well by functional connectivity, so by observing a frontoparietal theta connectivity that predicts participants' performance during the manipulation task. So once we have that, we have the metric. We confirm that theta activity predicts participants' manipulation abilities. We compared then 3 conditions. One condition, the participants do the task, but before each trial, it was just a black screen for 4 seconds. Then in the condition of interest, in the rotating condition, participants were stimulated with rotating objects before each trial. Then in the random condition, it was just the control condition with different forms flashing on the screen at 5 Hz. In terms of behavioral results, the behavioral results are quite clear. In red, you have the results without stimulation, in orange with rotating stimuli, and in blue with random stimuli. And we observe a clear advantage of the rotating stimulation protocol that are significantly better. So participants' performance is significantly better than for the no stimulation or than for the random stimulation. In terms of link between brain activity and behavior, then we extracted the phase locking between the visual stimulation, the rhythmic stimulation, and the brain activity to observe that frontoparietal connectivity, so those 2 regions that were following the stimulations, the more they were following the visual rhythm, better were the participants in the task. So we really have here an entrainment of theta oscillation in the dorsal pathway with visual rhythmic stimulation that predicts behavioral performance in working memory. But finally, and interestingly as well, is that some participants were trying to interpret or to deal or to manipulate the random stimuli. And what was interesting is that when they were trying to do that, their performance decreased. So it means that it's really the same network that is involved in both manipulation of information in memory and visual mental rotation. Because when the participants were trying to play with the random stimuli to try to create a rotation, we then observed that their performance was significantly decreased, as assessed by the negative correlation here. So to conclude, we observed with both types, we're using the same task, that both types of stimulation with rhythmic TMS in one node of the dorsal pathway or using optimized or information-based visual stimulation, we are able to induce stimulation, to induce behavioral enhancement that are related to the entrainment. So the next step here would be then to test other frequencies or the type of sensory stimulation. Can be auditory in the space, can be also other types of visual stimulation, and also to try to identify if this works on other brain networks than the dorsal pathway. And finally, we also want to explore the after effects of such stimulation procedures to try in the long term to define protocols to prevent cognitive decline or cognitive deficits in older adults, for example. So to conclude, I just want to thank my colleagues, and I thank you for your attention.